So we take a lot of the information from all of the mass spectrometry runs that you've been learning on how to process, and that data now gets curated and put into these online resources that we call the Atlas resource. And there's a number of different Atlas resources that make this up. And so this session here, we're going to talk about both the PeptideAtlas and another component called the SRM-Atlas. And so the two are quite distinct from each other. The PeptideAtlas is really a compendium of all of the different identifications, whereas an SRM-Atlas is we take discrete identifications that are well controlled, well characterized to become targeted proteomic assays. Now, this is where these two things reside in the back end there. There's a couple of other tools that we're not going to talk about, but at the end of when you've gone through all of your efforts of collecting your mass spectrometry data, doing all those statistical analysis tools, your protein inference, then you can store it into a relational database in which it becomes a PeptideAtlas. And that gets served back to the community. So all of our results, you can go and peruse and look at all those different applications. So when we use that, we know that at this one point where we've gone through our systems biology and trying to work out what are the biological questions we want to answer, whether it be biomarker discovery or some other component, and we've developed our technologies, both of the mass spectrometry and then also bioinformatics, it feeds into this point here. And that's where we can output these types of resources that can also not only be used as a repository to view the data, but also to use this kind of information to go back into applying these to answer biological questions and then improving on that technology again. So you can see how we've gone this whole full circle now, and now we take this information, we feed it back in, and so we can keep on utilizing these resources. And because we're doing things in an open source manner, a free manner, then you can also contribute and also gain benefit out of these types of analyses. So the PeptideAtlas is, this is the landing page that you'll see. And a lot of it is an effort by Eric Deutsch in the group and the people that are also working within the PeptideAtlas. This is the first page that you actually see, and you see there's also a number of different components that fit under it as well. And so you can look at very organism-specific compendia, whether it be chromosome-specific or other components. But it's a way that we can organize the data so that you can visualize all those identifications. And so the goal here is to try and define all of the mass spectrometry information of a protein. Not all peptides can make it into a mass spectrometer for some various reason, and not all peptides can be fragmented well enough so that they can be viewed by bioinformatic analysis and have the identification done. But what this does, it tells us all of the peptides that can be viewed by a mass spectrometer and can be identified by bioinformatic tools. This is an example of what happens when you build these peptide atlases over a number of many years in time. This is the human, what we call the human all peptide atlas, and so we bring together all of the human identifications that we have from mass spectrometry runs. And in each one of these bars, single bars, is an experiment. The red bar means this is new cumulative MSMS spectra being identified, and the blue bar is actually entirely new identifications of spectra as it goes on from each experiment to go through. And so as these blue bars accumulate, we get further and further and further into the whole proteome. But you can see here that we have these technology jumps. So each one of these experiments, you might see there's a relatively small number of identifications being done here. And this is in the early 90s, 2000, that we're getting into these big technology jumps when we go from, say, normal ion traps to these quadrupole type ion traps. And that's when you got these technology jumps. So you get a lot more peptides being identified and a lot more new peptides being identified which haven't been seen there before. And so we can keep on building on what we've got. But you can see here in the very recent experiments, mass spectrometry now produces very large quantities of identifications, many of which we've seen already before, but also produces new identifications which we've never seen before. And that's really the status of the technology as we are at the moment. But the key point here is with the transproteomic pipeline, we're able to control for our false discovery rates and make sure that at the protein level we're at the 1%, but at the peptide level that we're very much lower than that, and also at the peptide spectrum match level, it's very, very low. And that's really the type of effort that we're trying to do to make sure that what we present is actually correct. What does that give us? Well, when we look at a typical protein sequence, this is the protein sequence chain. These sections here is what peptides have been identified. And so you can see this peptide being consistently identified, this peptide being consistently identified, but this peptide here and there is not being identified at all. And that really tells you that there are difficult areas of a protein sequence that you cannot identify. But this kind of information gives you now signatures for that particular protein. And because we're using mass spectrometry, because we have limitations in mass range at the moment, until we get to these newer type of technologies that can do whole protein analysis, we're still looking at these fragments of proteins as sentinels or signatures that we can use to, one, identify the protein, but more importantly, two, quantify the protein. And one of these aspects is what we're also going to talk about. So Ulrika will talk about selection reaction monitoring. So selection reaction monitoring is really a mass spectrometry technique in where it's told what to do, rather than the mass spectrometer being programmed to do what it wants. And so here we have peptides going to the mass spectrometer. We tell the mass spectrometer in its first quadrupole to select out the peptide of interest by its molecular weight, fragment that peptide in the second quadrupole, and then read out each of the fragments in the third quadrupole to get a chromatographic peak. It gives us these advantages in terms of high specificity, high sensitivity, and also gives you this capability of being a multiplexed version of an ELISA. And so you don't need to have an antibody for this because the mass spectrometer is giving you that signature. And it's a molecular separation signature. We're down at the 1 Dalton level because of the resolution that we're here. We would like to be even lower, and there's techniques such as PRM, which enhances on this, but it doesn't really have the sensitivity at this point of the SRM, but it's getting close. But this gives you this quantitative peak that's measuring the peptide as it elutes off the column, giving you multiple points, and so you can get more accurate quantitation rather than just a single spectrum and say that's there. And so when we're talking about biomarker analysis, we want to have accurate quantitation of small numbers of peptides, but over many, many samples. And so we switch the point from a few samples and large numbers of peptides to now small numbers of peptides that we have as signatures for those proteins, but over many, many hundreds of samples. And then you can start to look at how is that protein behaving in so many different people. People that may be infected by a certain pathogen, or people that may be becoming sick from a disease. This is the type of analysis that we can use to monitor those proteins. And what you can actually do is then you can say, well, if I come up with a network of proteins, and here's a network of proteins that's got to do with glycolysis and glycerol metabolism, is that these proteins here, these enzymes, all deal with that molecule. And if you change the growth medium from one to another, you want to see what's the difference in all those proteins that are in that system. And so you can have signature peptides for each one of those, measure them as an SRM. As you change those conditions, then you can start to measure how those proteins change from each other. So this is a benefit of being able to do this in a very targeted manner and trying to look at a particular system at a particular time. And so in the end, you can come up with these things called these barcodes. So if you're interested in a suite of proteins that you want to measure in people's blood or urine or something else, you can create these assays that you have well-characterized signatures of these peptides, and it can be almost seen as a barcode. And so as you're measuring those proteins in a particular sample, it doesn't even have to be a human sample, it could be any other sample. If you want to measure, if you're looking for a pathogen in certain foodstuffs, you can create peptide signatures for each of those and just measure them over and over in time. And what you're doing is looking at that abundance change of each of those signatures. And so with that, it gives you a picture of really what we're trying to do is to create a resource now that we have both all of the peptides that are known for a whole organism, as well as utilize those in quantitative analysis. And so Dave will start off now with the PeptideAtlas.
Thank you very much. Can you guys hear me? Everyone okay today? It's been a long week. We're halfway there. So as Rob mentioned, the PeptideAtlas is a resource that has grown. Actually, it's grown over about the last 10 years. And it started out being primarily a collection of ion trap mass spectra. And it's now grown to encompass several sort of related resources, all built on a common infrastructure. I'll start you with a quote from the PeptideAtlas homepage, and essentially what this says is, it's a very large set of data collected from the community, processed in a very consistent fashion with as state-of-the-art tools as we can, with closely controlled FDR and error rates. So it's meant to be a resource for the community, and we do have feedback and interactions with other groups, such as NextProte or Uniprote, who take the identifications in PeptideAtlas and use those as evidence in their protein releases. So this is a quick outline of the talk. I'm going to give an overview of the PeptideAtlas, a little bit of history. I'm going to talk about exploring the PeptideAtlas, because as a resource, I think the most important thing that I can convey to you is what you could expect to find there. What good is it to you to go to the PeptideAtlas? Because it is meant to be a community resource. I'm going to talk a little bit about proteome exchange. So PeptideAtlas was first developed. There weren't any proteomics resources analogous to, say, microarray resources that existed. But now there is this thing called Proteome Exchange, where several large groups have come together and formalize the sharing of data and the submission and acceptance of data, and to the point where nowadays you almost have to submit your raw data when you write a paper. And also touch briefly on the process of submitting data to the PeptideAtlas and to the related resources. So the rationale for the PeptideAtlas, this is sort of from the past looking forward. The idea was that there were a lot of underutilized data sets out there. The tools were not standardized. People weren't doing model-based proteomics. They were getting a ton of identifications, but they had no idea which ones were correct or not. They were using simple thresholding. And so as a consequence, they probably had a pretty high FDR. And the idea arose that we could take all those data that were already sort of used and published and process them in a very consistent manner and get better control on the false discovery rates and the processes used to analyze them, and make a database that people could use so that it wasn't such a hodgepodge of different techniques. So yeah, the analysis software was not yet optimal, or sometimes people would run an experiment and find that it didn't really suit what they had tried to do, and so they essentially sort of save the experiment and move on. So the idea was, what further benefit can we extract from these experiments? And so here's a graphic depiction of, there's research group 1, 2, ... to group N. You have all these different experiments, and we'll do some sort of data analysis and validation, and we'll put it all in a database, or parts of it in the database that belong in a database, and build an interface so that people can explore this. Each PeptideAtlas build is the integration of a different number of mass spec experiments. Some are all from our institute, some are public. The bigger builds, like the organism builds, contain data from all over, and Z is actually constantly looking for new data, looking in PRIDE and Proteome Exchange to find new data sets to augment the builds that we already have. It includes experiments from all over the world. And at the time that the PeptideAtlas project started, ISB was very well suited because they were sort of on the forefront of proteomics technology. They were developing the profits and the sort of origins of the TPP. And so it was kind of a natural fit for them to develop the PeptideAtlas. So there's also some reasons why you would want to have one big resource that is analyzing everybody's data, because essentially that's going to be the most complete resource. And if you have data that's handled properly and it's as complete as it possibly can be, then you can do a lot of things with it. So you can validate predicted proteins, intron, exon junctions, validating various protein databases. So we regularly find peptides that don't map to one database or another. And as I said, UniProt and NextProt, which are, NextProt is sort of the next generation human form of UniProt, they're now ingesting information from the PeptideAtlas to link to their resource and to offer as protein evidence. We also view it as an important resource for experiment planning. People who are looking to do mass spectrometry can get information and insight onto what peptides and proteins might be observable, and they can also help to target their proteomics. And to some extent, these functionalities will be superseded by, or will be taken over by SRM-Atlas or SWOT-Atlas, but PeptideAtlas is the basis of this, and there are a lot of positive things that came out of it. It can be used as an aid in data analysis. There is a rich UI. It's maybe a little bit rambling at time, but there are a lot of different ways that you can sort of investigate your data graphically and in the context of external resources. And the idea is if you have a hypothesis, you can sort of walk through this database and drive yourself to some sort of knowledge, some sort of insight as to how you can proceed. And we, as I mentioned during the SpecTrust talk, we do have Spectrum libraries available, and that's also an important resource. And this validation of results is pretty important. There have been a number of papers that have come out who have claimed, some number of proteins or peptides seen, and upon subsequent analysis, it is found that, if you really look at it analytically, they're not as, that the original interpretation was maybe a little bit ambitious. So being able to put it in this pipeline and treat it like all the other data is useful in validating results in the literature. And Rob touched on this, so it's also helping to define the MSMS observable proteome. So some peptides are not amenable to mass spectrometry. It's difficult to purify them, difficult to ionize them, they're too short, they're too long. Some proteins are simply present in most samples at a very low abundance, and so you'll simply never see them. And so I'll show you evidence in the Human Protein Atlas, or PeptideAtlas, that we're sort of approaching a plateau in the number of proteins that we can detect, even though the number of peptide identifications are still increasing. And so we're homing in on this idea of an easily sort of shotgun MSMS observable proteome. It may be that with techniques such as SWOT or SRM, we can go deeper into the proteome, and Ulrika will touch on that a little bit later. So these are the different components of the PeptideAtlas. So the TPP and the PeptideAtlas are tightly integrated. I'll show you a slide in a sec. So basically, the TPP is used to process data going into the PeptideAtlas. The PeptideAtlas, in turn, generates requests for new features and ideas and sort of tests things at scale that help drive the development of the TPP. And so we view it as a cyclical and symbiotic relationship. So the TPP is here, PeptideAtlas. And more recently, so again, PeptideAtlas is MSMS datasets, mostly ion trap, but other types as well, QTOF. SRM-Atlas, as Ulrika will talk about, as well as PASL. And closely related, so SRM-Atlas is a database of SRM assays. SRM QuantAtlas is a database of SRM assays that have actually been done in calibration curves. So we have limits of detection, limit of quantitation, and we can make these plots. And these are sort of much more characterized SRM assays. It takes a lot more work, so this is less complete than this. And the SWOT-Atlas, I think Sam will talk a bit about later today, and that's using this new technique called SWOT, or relatively new, or data independent acquisition. And they're built on a consistent framework in that they're sort of in a shared database, and they use some of the same tools. These are some of the funding organizations. At the beginning, the opening session, there were people from HUPO, Josh LeBaire and Mark Baker. Also, Rob is a vice president at HUPO, and then an affiliated project, the Human Proteome Project. And Eric's also very active in the Proteome Standards Initiative. So this is a schematic of the PeptideAtlas workflow and sort of the ecosystem in which it exists. So essentially, people do shotgun experiments. People in the community do shotgun experiments. They submit them to PRIDE or NIST, or as it turns out, another new resource called Massive, which are part of Proteome Exchange. And so there's an exchange mechanism whereby things get propagated. If you submit to one of the raw data repositories, it gets propagated to Proteome Exchange and gets propagated to the others. PeptideAtlas also has a raw data repository. It's not as extensive as PRIDE because we're not primarily a repository resource, but we do have a number of important data sets that have been used in our builds. We do sequence and data and spectral searching, run everything through the TPP, and as Rob showed you, I don't know if you appreciated it when he showed that slide, but we use very, very stringent FDR cutoffs at the PSM level, so that at the protein level, especially when we're combining all these data sets, we still have a manageable FDR. We typically aim for a protein FDR of about 1%. And that is actually learned from the data set. We do a decoy estimated FDR, and so we have a pretty good idea of what our FDR is, and think that if anything, it's a little bit conservative. And then the PeptideAtlas has various ways of contributing back to the community, and so it's sort of again a cyclical feedback type of situation. As you'll see when you're going through the tutorials, most of the pages where you have different peptides, you'll have a link that has this stylized PA, and that means that it's a link to the PeptideAtlas. It typically drops you into this search page, and if you'll see, there's a percentage sign on each side. Those of you who are familiar with SQL or SQL, the percentage is the multiple character wildcard, and so when you're driven over here from this peptide, they basically give you, by default, a wildcarded search. You can also take those away and search for just the peptide of interest. Looks like these are all the exact peptide that was observed here. And what you see is, so the search sort of executes right away, and what you see is you've got some hits in human, and you've got at least one hit in mouse, and I can't tell what's going on down below, but basically your results are organized like this, and then you can click on one of these identifiers. If you search instead for a protein identifier here, you'll get essentially a list of protein accessions, and you'll also get a list of synonyms. So as you know, there are numerous databases. We use Ensemble, IPI, Uniprot is our main one, Uniprot, Swissprot, Nexprot, and so we have translations to all those accession spaces within PeptideAtlas. So if you look for one, you will often get results in others, and so you can compare and contrast. So I'm going to go over a little bit of an exploration of the PeptideAtlas. Again, my goal is to let you see what there is. Obviously, any questions, please let me know. As Rob showed, this is the landing page for PeptideAtlas. We have some links to other resources, including the resources within PeptideAtlas. We have a big button here to submit your data to PeptideAtlas or the related resources. Actually, at least one person in this room has used that submit button to submit some data to the SWAT Atlas. So I view the PeptideAtlas as sort of a set of resources, or I mean, for me, I know if I'm looking for something, I know where it is, but I know it's hard coming into a website that has a lot of links and a lot of words on it that, you know, what is really there. So at the most basic level, we have a number of raw MSMS data sets. Some of them are from our lab, some of them are from the community. We also have a modest number of spectral libraries, but for our bigger builds, we're actually planning to generate these libraries, and we just haven't got the infrastructure in place yet. But we think that they will be, in terms of breadth, in terms of the number of peptides that are in these libraries, they will probably be bigger than the NIST libraries, but right now we're not making them as tightly quality controlled. But we think that it would be a good augmentation for the NIST spectral libraries. There's a number of ways you can pull out high confidence peptide and protein lists. So if we have a build for an organism that you're interested in, then you can easily go and get information about the peptides and the proteins that we've seen in the various builds. And we have a lot of examples of integration with external resources, and I think that's really important because, I mean, there's a lot of effort going into resources around the world, and to be able to tightly integrate with other resources sort of makes each one more valuable than it would be in isolation. And it is a pretty rich exploration environment. It may take some getting used to. I will go through some pages here, and we may do a tutorial where we take you through a few of the interfaces and let you click around. But if anyone that's curious, I encourage you to just go and start looking around and see what you find. So the first couple things I'm going to talk about are essentially large-scale access points to the PeptideAtlas. So this is the data viewer. This is the raw data repository. Each one has a PeptideAtlas experiment accession number. It's got a little widget here that you can enter some text and then search and limit that and help you find different things that you may be looking for. These are all downloadable via this. You can click each section and open it up, and you can get download links, and different of these data sets have different file sets available. But there are over 1,400 different experiments, and again, some have the raw files. Almost all of them have search results, and we have them for quite a variety of organisms. As I mentioned, we have spectral libraries. Here's some that we offer on our own. We have a number of NIST ones that continue on down the page. And you guys yesterday went through the first part of the Proteome tutorial, and you saw the page where you could see the spectral libraries, and that's essentially querying this page or a similar page and finding out what's available. And so that's, as we add more, then those will show up in Proteome. There's also, so I haven't actually introduced a build yet, but we have these builds. So each time we take a collection of data for a particular organism and make one data product, that we call that a build. And we have them for quite a number of organisms, and we make them available in various data formats. So they're available as flat files, which would be a TSV or Excel compatible file. Get them as XML, which may be a little bit more parsable programmatically. We also export them as what's called a MySQL dump. So MySQL is a relational database. It's free, and you could take this and basically download this MySQL file and generate your own database, and then you'd be able to interrogate it as you wished. And again, spectral libraries are associated with some of these builds. So actually, one more thing that I will say here, there's also, we have some, what I'm going to take you through now are more is more of an exploration. You have one protein or set of proteins in mind, and you're sort of navigating through links. And that's a manual process, and it's not that useful for a lot of applications. We do have a number of query forms where you can enter in a list of proteins manually, or you can search for certain modifications, or you can you have a file of protein accessions. You can upload and search those, and basically get down subsets of these large data sets that are here. So basically, you don't have to get the whole results file. You can get subsets of them that are tailored to your needs. So a little bit about builds. So this is our builds page, and basically it continues on down the page. I'll show you a little bit more later. So whichever one has this little dot by it means that that's the one that's selected, and you can select it here, or you can select it on a number of pages. It tells you the number of distinct peptides in that build, the organism, and then a brief description. If you click on any of these links, say I clicked on that human 2013 build, 2015-03 build, and this is actually, so you see some information to build, the description, and the reference database. So this is actually a little bit interesting. So we make a composite database when we search in order to be as complete as possible. We then take that composite database. So we know from the past which proteins we've seen. And so we take essentially the uniprot, swissprot portion plus tremble, which basically makes the uniprot complete proteome. So they have different definitions of what the proteome is. The swissprot component of human is just over 20,000 proteins with tremble and swissprot and the var splice, the isoforms. It's on the order of 57 or something like that. But we then map it to all these different databases. This nextprot SNP file is interesting. Earlier we had a question about SNPs. So what we do here, so a couple different databases annotate SNPs. So they give you a primary sequence and they say, okay, at amino acid 33, that alanine can be a valine or a threonine. So what we do is we take that and we encode that in a peptide with some context and put it at the end of each protein. And what it enables us to do is actually search for all these variants. So in the uniprot file, they're called variants. Some of them are from dbSNP, some of them are from other places, primarily from cancers and other things. But basically, by including that in our search database, and then again in our mapping database, we are able to detect these, and we're able to display them in our user interface. And so if you're interested in variants, in SNPs, especially for the organisms supported by uniprot, we can show them. And this is now, for the human and the mouse, this is done, and it's becoming the norm for other builds as well. So there's some more information. Basically, it gives you an outline of the last two builds and lets you know how many new data sets, how many new peptides. Here we've added a number of data sets, but the number of peptides hasn't gone up that appreciably, and the number of canonical proteins has actually gone down. And I will touch a little bit on this idea, but it has to do with what Luis was discussing yesterday in protein profit and protein parsimony. So if you look at all the peptides we see with high confidence and you map them to proteins, you will actually say, okay, we map to 17,000 or something proteins. But some of those proteins do not have unambiguous evidence. And so this is a very conservative view. These are just proteins that we've seen with at least 2 peptides. Each of the peptides have to be greater, or 9 amino acids or greater. So we don't even include shorter peptides. So it's a very rigorous look at how many we've seen. So I'll show you evidence that this number has, in essence, plateaued. This shows you the different samples that are in the build. And as Rob showed, you see this plot where you see the increase. So actually, the magnitude at any point in time here is a cumulative number of distinct peptides we've seen. And right now, it's somewhat over 1 million. The blue bar represents the number of peptides observed in this experiment. And so a peptide, or an experiment that has a high bar, that's just the number of peptides seen in that experiment. The number of new things can be calculated by looking at the difference between this bar and the previous bar. And one other thing, so you see this one here is actually kind of fat. The width of this bar indicates the number of spectra in that experiment. So this guy is kind of fat, and the blue is rather short. So they didn't see that many peptides out of this experiment. But if you go down here, these are very thin, and yet they have a high blue bar. So these new experiments represent what Rob was talking about, which is technology improvements. I mean, these are older data sets, and things are working better. We still do see these plateaus, but then these inflection points where technology has improved and we're able to look deeper into the proteome. Also on the builds page is this heat map, and what this is showing you is basically for the top 25 proteins in this build, what is their distribution across the samples. And in some cases, it's interesting. I mean, like, For instance, this particular sample has this protein or these two proteins expressed at a high level. Sometimes, so these may be synonyms. You can see the accession spaces are different. This is an SGD accession, and this is a uniprot accession. So it's very likely, since they're in lockstep, that they're the same thing. But this sort of gives you an overview of which proteins are most highly expressed. So this is another slide showing the builds page that I showed earlier, but blown up a little because that was too difficult to see. And you can see that we have some 21 organisms, I think, where we have a sizable build. We're, again, using very low FDRs. This is 0.002%, or 0.0002 as an FDR threshold at the PSM level. And what the target is, this will almost always yield a 1% FDR at the protein level. And so what we essentially do is do the build, get to the protein level, look at the number of decoy proteins that we've seen, and then calculate the PSM level required to get that protein. That's why they vary. We don't have one fixed amount. And it really is dependent on the data that we're using. This is the number of samples, so the human is clearly the biggest right now. And we definitely have more samples than this, but as time goes on, some of the smaller ones aren't really worth our while to bring them along. They're lower mass accuracy, things like that. And in addition, within just the human, we have all these different builds. She mentioned the human brain has a relatively modest FDR here. It's a smaller number of data sets, and because there's this amplification of error as you get bigger data sets, at a small number of experiments, you can actually use a higher FDR at the PSM level, at the matching the spectrum to this peptide or this peptide ion. You can use a lower or a higher PSM FDR and still yield a 1% protein level FDR. And this tells you the number of spectra and the number of distinct peptides and the number of proteins covered. So this is kind of the interface that most people will see when they first go into the PeptideAtlas. So this is the main search page, and as you can see, I've entered in, this looks like a gene name, and I've gotten some hits down below. And you see that within human, we've seen a number of things. So Ensemble actually has this thing represented numerous times. So that's one. Ensemble is much bigger, but they represent isoforms as distinct accessions, and they much more often have completely identical sequences. And so if you get a hit to an ensemble protein, you typically see a lot. We normally use with the uniprot ID, and so if you were to click on that, you'd get a protein page. And the pages are set up into sections. You can close and open sections using these little tabs. It tends to be, it is sticky in the sense that if you close all the tabs and you go to a new protein, then they'll remain closed, and then if you open one, then it'll remain open. And basically, we just have some information, essentially cross-referencing to all these different resources. The protein group is analogous to the peptide profit protein group. This is a canonical protein, so this means that there is unambiguous evidence for this protein. We have a number of Go terms for biological process and cellular component, and then link basically map to these different accessions, and we show that it's on this particular KEGG pathway. And this is a depiction of the peptides that we've seen in the context of the protein sequence. And you can see that we've seen virtually the entire thing. In between, there are these little nibs that are called unlikely. So each of these things is called a track. So this is the observed protein track, sequence position track, and you can see that it's just over 1,000 amino acids. There's a predicted anchor sequence, and then we've seen all these sequences. And these ones that are unlikely are generally because they're too short or too long. And too short is 6 amino acids or less, and too long is, I believe, 50 amino acids or more. And so for all intents and purposes, we have seen everything from this protein that we're going to see. You'll probably notice that these things are kind of a nested set. And what that means is, in addition to, well, first let me say that there's a color key. So these are observed peptides with a single genome mapping. So basically, these peptides map only to this protein. In some cases, if you have a member of a homologous family or a closely related family, or other things, you will see, or a gene duplication, you will see that there are ambiguous genome mappings, and then there's no color. But the darkness of the color represents how many times it's been seen. So the darker ones have been seen more. And so what you'll see is you have these nested sets, and basically what this means is a lot of these are semi-triptych or have missed cleavages in them. But typically the ones that you see the most are fully triptych. This is a little bit further down the page, and this is a different view where all the red sequence has been seen, and you can see the different peptides. And these peptides are broken out as triptych. So this you can view this as triptych, or you can view it in the sort of normal, 10 by 10 or 10 amino acids as a block kind of view. But this shows again that we've seen almost all the triptych peptides, and the ones we haven't seen are just too short, or in some cases too long. And it looks like it's not 50, maybe 30. This particular protein is interesting because we've seen a number of SNPs. So I was talking before about the variant sequences that are in our database. So in this case, we've actually seen 4 different SNPs. Some of them we've seen at a very low frequency, 1 out of 360, 3 out of 300. But in this case, we've seen this all 53 times we've seen this peptide. It's been in what was considered the variant form in UniProt. And we've actually submitted these to UniProt and gotten them to switch, essentially, the polarity of the SNP. Because when they were doing the SNPs, they took a number of individuals and identified it and sort of arbitrarily said, well, yeah, this is what should be in the population. And obviously, different populations vary. But basically, in our data, and we have, and this is over 1,000 samples, all of them are this. And so probably what we're seeing is really should be considered the canonical, and the other should be considered the variant. All the SNPs are depicted here. We basically bubble the ones that have been seen up to the top, and so each SNP gets a number that is reflective of its position in the sequence as you go along. And so basically we've put SNPs 1 through 10 at the top because those are the ones we've seen, and then we start again with SNP 2, which has not been seen, and you can see that these 2 amino acids, they're black, and so they have not been seen. So this is a sequence context view of the peptides that we've seen that also includes the SNP peptides. And you can see that, yeah, the canonical is considered to be alanine, and we've seen it only as a glycine. Below this, you see an exhaustive list of peptides. I don't think this is a particularly good example just because we've seen so darn many of them. Here is a slightly different example. If you take one of these peptides and click on it, you'll get a peptide information page. So the previous page was information about this protein. You can look at any individual peptide. So this particular peptide maps to multiple accessions. You can actually click this and get a clustral alignment of them. I will not show that. You can see all the different proteins it maps to down here and whether or not it's a variant mapping. So the one we were looking at before would have showed as is variant for this protein. Down below that, we have consensus spectra. So we basically report all the peptide ions. So these 3 have the same sequence in that they're not modified, but they have 3 different charges. Then going down here, we have different mass modifications, combined with different charge. So again, the combination of linear sequence plus mass modifications plus charge represents a peptide ion. So for these ones that are unmodified, we actually have consensus spectra, and you click on one of these and you'll see a spectrum. But we also have all the spectra that were observed, and this is a plot that shows the observation frequency for each one of these. But if you click on one of the spectral links, You'll get a spectrum in what is called the lorikeet spectral viewer. You can see that this has a nice B ion series, a nice Y ion series. You can modify these things, and it's actually quite a nice dynamic viewer. And this is another protein example that I think is a little more interesting. So the previous one we saw, we just had a ton of peptides all over the whole protein. So this one, the peptides that we've seen are restricted to this end of the protein. It's a much shorter protein, but you can kind of see why. So basically, what we have is predicted transmembrane regions that are going to be difficult. Basically, you often don't see transmembrane peptides because A, they're hydrophobic, difficult to purify, and B, there's not a lot of basic residues, lysine and arginine, so they don't typically have tryptic fragments. We don't only have tryptic peptides in the peptide atlas. We have a number of other enzymes, and it actually adds a lot of diversity. But in this case, this is the unlikely theoretical is based on tryptic peptides. And you can see that this gap here is also explained because there are a bunch of tryptic sites in through there. And so a lot of cases, you have a much more restricted set of peptides, and you can explain it based on other biological information. We also have PTM localization, and I think this was as part of PTM Profit. And it actually seems a number of you guys are quite interested in phosphorylation. This is a little bit hard to grasp right away. So this is a serine and a threonine and another threonine. Just looking at this plot, which do you think is which is the best one, if you were just to look at this? The serine? So even though the height of this actually tyrosine is higher? So you're absolutely right. So basically, in PTM Profit, what you saw was for each site in a peptide, a single observation, what the probability was. And so the height of these bars represented probability. And so in that case, the highest bar was the best probability, and that would be the best one. This is the summation of that information across all the observations. So you see here that there's a 131 on top of all these. Basically, there are 131 observations. And for 119 out of those 131, the probability given to this threonine was less than 0.3%. So the fact that this is a high bar is not important. What is important is that it's a low probability. Same with this one. It's even less convincing. Although here, this serine, 34 of them are greater than 0.9, and 76 of them are greater than 0.98. So the sum of these two are the number of observations above 0.9. So clearly, in this case, we have strong evidence that it's this serine. As you'll see later, the phospho build is actually quite big, and so we've covered a lot of phosphosites. And that's not just because we've done a lot of phosphoproteomics, but rather because a lot of people in the community have, and we've obtained those data sets. And those are the ones that we have data for in this peptide atlas. And you can click on that and go to essentially see those things within the context of the peptide atlas. The ones that are yellow, we don't have any information for. And in some cases, there are boxes on the KEGG map that are clear, are without color. And those are proteins in whatever organism. Okay, so right here. So for whatever reason in this organism, that is not represented, or we were not reported a protein accession that represents that particular entity. And for some of the lower organisms, it's much more common to see empty boxes. Another sort of mass way that we can view data is through so this is a HUPO initiative, the Human Proteome Project. So they have both a chromosome-centric initiative as well as a disease and biology-centric initiative. And so we have a number of groups, about 10 so far, that have submitted these curated lists. And in a lot of cases, they're backed by a publication. In this case, it's quite a large list. There are 1,172 ovarian cancer-related proteins. And so what we do is we provide this list, and you can essentially take that and search either SRM Atlas or Peptide Atlas right away, or you can use this widget and essentially start to type, and that will limit the list. And so at this point, by just typing in CAN, on the way to cancer maybe, we've gotten rid of all but 14 of the whole proteins in this list. So if you have some proteins or protein family you're interested in, you can type in the name, you can select them, then you can erase the text and type in something else, and this is a pretty powerful interface for building subsets of these lists that are interesting to you. And again, this is an example of how we've integrated with information that's in the community, and we try to basically make an affordance so that you can use this to interrogate the Peptide Atlas. This is another resource. There's a group who did sort of an all versus all BLAT. They came up with ortholog groups across all different, a huge number of species. We've taken out the ones for which we have PeptideAtlas builds, and basically this shows a cluster alignment of those proteins that are related, and I take their word for it that these are related. I mean, obviously these ones down here don't seem as closely related as these ones up here, but then you see across each of the build how many proteins we've seen. For the chimpanzee and the dog, we haven't seen any, so it's not shown in this list. So now I'm going to talk a little bit about specifically a couple different builds, and this is a timeline of new data sets. So the human atlas was pretty big back in 2013, but there have been a number of big data sets from Akhilesh Pandey and from Kuster, and there's a CPTAC study, and basically this from Hamid Mirzai. He had a big data set that used a bunch of different enzymes, so he actually added a big number of novel peptides to the build. So anyway, as we go along, we get these builds, and these are pretty high profile and interesting builds, and so we're very lucky to be able to incorporate them into the human atlas. And you see here that some of this data was used for the human phosphoatlas. And again, the width represents the number of spectra, the height at any one time represents the cumulative number of peptides we've seen, and the height of this blue bar represents the number of peptides in that experiment that were over our threshold. And this barometer over here, the colors don't really represent these colors. This basically says that with the canonical proteins, with the canonical, so with our conservative view, we've seen 70% of the proteome. 133 million PSMs, a million distinct peptides, and this is a very low PSM FDR. So actually, I misspoke before. That was a peptide level FDR. So this is 0.0009% PSM level FDR, simply because there are so many proteins in here. And again, it yields a 1% protein level FDR. And this only considers peptides that are greater than or equal to 7 amino acids. So this is illustrates the plateauing effect I was talking about. So over the last, that timeline, you saw that we had a number of data sets contributed. And so the number of peptides has actually gone up quite a bit from around 400,000. It's like 1.5 or 2.5 times higher at just over 1 million. At the same time, the number of proteins we've seen has not increased that much. And sadly, essentially what that means is that the additional peptide hits we're getting are to the proteins that we've already seen. And this is unfortunate, but what we hope is that as technologies increase, then we will hit another threshold, and we will basically be able to increase the coverage that we have. I'm not going to delve into this too much, but basically what we're looking at is how fully covered for the latest human peptide atlas, how fully do we cover the NextProt complement of proteins. So NextProt is essentially UniProt without things like HLA or immunoglobulins, some very highly redundant or very highly variable proteins. And you can see here that there are different protein classes. So canonical basically means we have unambiguous evidence. It has more than 2 distinct peptides that map only to it, and they're greater than 9 amino acids. And so that is a very rigorous criteria. There's these other that are ambiguous. So some that we have some evidence or weak evidence, and that would be things like shorter peptides or insufficient evidence. And I don't honestly, I don't fully understand all the subtle differences between these, but basically this one is certain. This one is ambiguous. We have evidence, but we're not sure it's actually that one, or we're being too conservative to assert that that's correct. Redundant means that we have this peptide, this protein has been implicated, but we have evidence that all the evidence that points to that has been seen in other proteins. And then finally, the ones that simply were not detected. And that's only about 16%. So the ones that were not observed, so that you could actually look at that as sort of the non-covered proteome. So that would give us about 84% coverage in some fashion. This is a breakdown by chromosome. So NextPro provides a list of accessions and chromosomes. And you can see for the different categories, canonical, uncertain, redundant, or not observed. We have pretty good coverage. So the canonical coverage for most of them is around 70%. There's a few outliers. The Y chromosome, we have very few, things like that. This is the human phosphobuild. You can see that simply by the width here that we have far fewer spectra, and we have far fewer number of peptides observed, about 1 order of magnitude. So the current human all has about just over 1 million, so that we have about 10-fold lower. And we've covered about 50% of the proteome. This is from a relatively modest number of samples, purified by different phospho enrichment methods. And consequently, this PSM FDR is a little bit higher. Is it? Yeah, it's a little bit higher because it is a smaller number of experiments. But if you think about it, I mean, 50% coverage of phosphopeptides is pretty startling. This depicts the growth of the plasma peptide atlas. So the plasma peptide atlas is a subset atlas comprised only of plasma experiments. What this shows is, in essence, a technology jump. And so this was the previous build in 2010, and it was pretty modest. And then basically we went off and got these 2 big data sets from IPAS and Roche, and that essentially gave us 5-fold more total peptides observed. And in fact, this is where Roche ends, and it's gone up even further since. And in the entire plasma build, we've seen about 21% of proteins. The plateauing that we saw in the human all has not occurred yet in plasma, and so we're hopeful that we can see deeper into that tissue type. So I'm going to talk briefly about proteome exchange. There weren't any raw data repositories before. Now PeptideAtlas is somewhat of a data repository processed through TPP. We make the data available for download and builds available for browsing. PRIDE, sponsored at EBI. Protein identifications are the main focus. They accept submissions from the community, but they don't reprocess the data. And so the quality of the data sets will vary because the quality of the analyses is varied. There was an initiative called Tranche, which was actually very interesting. It was a distributed file system like Kazaa. And basically, the idea was that every institute would make some disk available, and the files, all the MS files would be stored as distributed blobs, and they could be called, and there'd be redundant, and it was great until it wasn't. And basically, there was, at some point, there became corruption in the thing, and so a lot of data that had been deposited couldn't be retrieved intact. And so that is now defunct, and it's being replaced with an initiative called Massive. And Massive is planning for these eventualities in advance, and the hope is that it will be a more robust repository. And there's also was an initiative at NCBI called Peptidome, which was terminated. Funding was terminated a while ago. And yeah, Massive is now superseded Tranche, and GPMDB, apparently there's a quote on their website saying it's not a repository, and they reprocess all their data with Xtandem. So more recently, there's been an initiative to have a consortium of groups that share their data, and this is called Proteome Exchange, and PRIDE and PeptideAtlas, and this is the logo for the massive group. I couldn't find a logo for them. And there are data sets there, and they have rules for sharing data between the member parties. And that's actually where we get a lot of our new data for PeptideAtlas builds. They have a number of data sets for download, and this is a graphical depiction. So the idea is that journals will require submission. These different repositories receive data, and Proteome Central acts as a broadcasting mechanism to send things out as needed. Oh, and this was named Dataset of the Year, PDX 13. So finally, I'm going to talk just very briefly about submitting data to PeptideAtlas and the related resources. As I mentioned, there's this big submit button. You can push it, and you'll be brought to a page. You can fill in a username and a password. You can basically say when you want the data set released. So when you submit here, it's not automatically public, so people can't see it. But there's a mechanism where, and you essentially upload your files. There's a mechanism where you can set up a reviewer access. So you can make your data private, submit it, and then allow reviewers to come in and take a look at it before publication, so you haven't sort of violated the pre-publication clause. And the hope is that that will encourage people to upload data, because one notion in proteomics is that it's important to have all the data available, and journals seem to be on board with that now. And like the PAE experiments, we have these pass accessions, and this represents all the publicly available pass accessions. And there are different types. So some of them are SRM, some are MSMS, and some are SWA. And so this is a resource for data sets as well. And that's all I had.